Good afternoon, everyone. I'm Dr. Adeeb Al-Zoubi from Jordan, co-founder and chief scientific officer of MSI, founding president of Stem Cells Arabia and Amman, Jordan, and a co-inventor on the COVID Hunter. Today, we are witnessing the birth of a new technology that will change how we detect SARS-CoV-2 that causes COVID-19. You see, one of the biggest challenges we have in controlling the spread of COVID-19 is that the virus has a very high rate of infection, while the detection methods, on the other hand, are old, such as PCR, are old, slow, expensive, and require tedious lab work. We at AMSI and Stem Cells Arabia aimed at attacking the virus by employing an immediate and highly sensitive detection method to deprive the virus of its high infection rate. So we are hitting the virus at its strongest point. The COVID Hunter is a new technology in the detection of SARS-CoV-2 that causes COVID-19. The science of the COVID Hunter technology is elaborate, breakthrough, and revolutionary by all means. This patent-pending technology utilizes a unique combination of light waves, sound waves, combined to hone in on specific physical, chemical, and biological characteristics of SARS-CoV-2. So we are basically surrounding the virus and characterizing the virus on all its characteristics all at once. Through focused research and tireless work, we at AMSI and Stem Cells Arabia analyzed and specified these physical, chemical, and biological characteristics of SARS-CoV-2 and used these characteristics as one single value to target the detection by the COVID Hunter. The sum of these specific SARS-CoV-2 characteristics is not found in any other virus or any other target and constitutes a unique thumbprint of the virus. So this is a unique thumbprint of the virus that is not found anywhere else. We use that to be detected by the COVID Hunter immediately. By using safe electromagnetic waves, including radio waves like those used in car remote controls and infrared waves like those used in TV remote controls. So basically this unit here uses these two waves, radio waves and sound waves. Both are safe. By using infrared waves and radio waves combined with a proprietary method for detection, we have been able to successfully detect SARS-CoV-2 in a safe, instant, and specific manner. Safe, instant, specific, and more important than all, non-invasive. I don't need to take a sample. In a way, to think about it from a different point of view, the COVID Hunter visualizes or sees the virus in real time and gives the result in real time. You point, press this button, it beeps, virus is detected. That's it. That's how fast it is. You don't need to wait minutes for chemical reactions to take place. The COVID Hunter technology was developed by a group of international scientists, researchers, engineers, and inventors, led by a Jordanian team. And here I want to take this opportunity to sincerely thank our inventors team for this great world-changing achievement. Thank you all very much for this great work, guys. The technology is owned by MSI and was developed in collaboration with Stem Cells Arabia and Amman, Jordan. This technology is safe, accurate, fast, inexpensive, and easy to use. Inexpensive because you can perform thousands of scans per day. You only need to change the battery. That's all. No consumables to use here. The COVID Hunter can easily perform up to 300 scans per hour. That's a lot. That's very fast. Just the time to scan. 5 to 10 seconds per object or person. This allows the scanning of millions of people daily by using 1,000 devices only. What does that mean in real life? You need one of this, one COVID Hunter device only, used at a school, or at a business, or a company, or a restaurant. You can literally open that school and scan all incoming students every morning and keep this school or business COVID-free environment. Those who are infected can go home, small percentage, but the rest can go to school. So we can literally open schools and universities and restaurants. On a larger scale, we can scan the whole state of Michigan or New Jersey, for example, with about 10 million population each, in less than a week. Less than a week. Can imagine the whole state of Michigan will be scanned less than a week using only 1,000 devices. We can scan the whole country of Jordan. The whole country of Jordan can be scanned in less than a week using only 1,000 devices. Let's go larger a little bit. If we have 100,000 units, only 100,000 units of this, in place, in operation in the US, we can scan the whole population of the United States of America in 1 week. More importantly, we can identify those people who are infected with the virus and the other part, the other percentage, the other small fraction of those infected who are infective. So we can differentiate between people who are infected and people who are infective. All of this can be performed without taking any sample from the body or performing any lab work. Dr. Anthony Fauci once simply explained that not everyone who is infected with the virus is infective. What's the meaning? What's the difference between the two words? In other words, not everyone who catches the virus is a transmitter of the disease. So you can be infected, but you're not infecting others. Or you can be infected at the end of your infection, then you become non-infective. The COVID Hunter identifies infective individuals who are spreading the virus from those who are infected but are not spreading the virus. What does this mean in real life? Identifying the small percentage of society who are infective individuals makes it easy to isolate them and stop the spread of the virus. We can literally stop the spread of the virus, while the non-transmitting infected can go back to normal life, go back to work, go back to school. Another level of complexity that we encountered with this pandemic early on, and we know it, and everybody kept hearing about it, is that a significant percentage of people who are infected with the virus are asymptomatic. Asymptomatic people who are infected but don't show any symptoms of infection. These people may be spreading the virus to healthy individuals without being detected, especially in the first few days after infection, where they cannot be detected by PCR. If you perform PCR tests on them, they can be PCR negative in the first few days, but they still have the virus. The COVID Hunter can be used to identify these silent spreaders. We call them silent spreaders of the virus, allowing us to focus on stopping the spread of the virus before it reaches new, healthy individuals. On another level, after 1 year, we've been into this pandemic almost 1 year, right? After 1 year of human suffering, loss of lives and livelihoods, the only hope that we have today is vaccines. That's all what we got, vaccines. Vaccines work on improving human health by preparing and priming the human body before the actual infection takes place. The COVID Hunter does not compete with vaccines. The COVID Hunter complements vaccines. How is that? If it's a pyramid of people who are infected, the base of the pyramid represents people who are incoming as new infected individuals. At the tip of the pyramid is the small percentage of people who are getting very sick and are susceptible to developing complications. Vaccines represent the tip of the pyramid. Our device represents the bottom of the pyramid. While vaccines work on the human immune system, the COVID hunter does not work on the immune system. The COVID hunter works on the other side, on the virus itself, by identifying and stopping the virus from reaching and infecting new human targets. So we cut the base of the pyramid. No influx of new recruits for the virus. So you deprive the virus from reaching its new targets. Therefore, I don't have new cases every day. I'm just trying to get rid of the people who are, or trying to heal the people who are currently infected. Vaccines aim at decreasing the rate of death and decrease the incidence of complications in people who are infected with the SARS-CoV-2, whereas the COVID hunter aims at decreasing the rate of infection with SARS-CoV-2, thereby decreasing the number of new infections. Now, detection is the major problem with this pandemic. Regarding detection, there is a major difference between the COVID hunter and other methods currently in use for the detection of SARS-CoV-2, such as PCR and antibody tests. While PCR and antibody tests require sampling, performing chemical reactions in the lab, and tedious lab work, the COVID hunter is based on the immediate, direct detection of the physical presence of the virus inside the human body. So you're detecting the virus inside the human body without touching the human body. From distance, you can click and you can detect the virus if it's inside the lungs or inside the throat or inside the nose or the nasal or the different areas of the body. If SARS-CoV-2 is present within the area scanned by the COVID hunter, an immediate signal will be heard or seen by the user indicating the positive identification of the virus. An additional and unique feature of the COVID hunter is the ability to identify and detect the virus outside the human body on contaminated objects such as door handles, mobile phones, and other objects. Why is this important? It allows us to immediately spot and identify and disinfect these objects effectively and properly and quickly. So during the past 9 months, our teams at Stem Cells Arabia and AMSI carried out extensive and elaborate series of experiments to demonstrate the specificity, sensitivity, and accuracy of the COVID hunter to detect SARS-CoV-2. So this device was put to the test extensively during the past 9 months. We manufactured and utilized several COVID hunter prototype devices to assure consistency and reproducibility of results obtained by these different devices. We then performed 2 types of testing. There are 2 types of testing: testing of samples and testing of humans. First, the testing of nasal swab samples was carried out at a licensed diagnostic laboratories certified by the Ministry of Health in Jordan to perform specialized COVID-19 testing. These tests aimed at measuring the accuracy, sensitivity, and specificity of the COVID hunter prototype devices compared with PCR technology that's available for this test. Second, our team was granted an institutional review board called IRB approval from the Ethical Committee at the Royal Medical Services in Amman, Jordan, which is the highest medical facility quality-wise in Jordan, to perform testing on confirmed COVID-19 patients who are residing within the quarantine areas and in hospitals. So this is live test on patients. We performed thousands of tests on confirmed swab samples and on infected as well as healthy individuals, and the results were outstanding and were submitted to the US FDA here as part of the EUA application, or Emergency Use Authorization application, to approve this device for use later on. And here are our results. To demonstrate that the COVID hunter specifically identifies SARS-CoV-2 but not other viruses, we tested positive samples of different viruses, including SARS-CoV-2, the one that causes COVID-19, SARS-1, the old one, MERS, Influenza A, Influenza B, HIV, and dengue virus. the COVID hunter detected only SARS-CoV-2 positive samples and none of the other viruses, showing 100% accuracy to detect only, 100% specificity to detect only SARS-CoV-2 positive samples. We then tested more than 4,000 swab samples and compared COVID-19 results, COVID hunter results with PCR results. All PCR positive samples were detected by the COVID hunter as positive. So this device positively identified 100% of the positive samples as identified by PCR, showing 100% accuracy among PCR positive samples. On the other hand, this is addition. 4.8% of PCR negative samples were identified by the COVID hunter as positive, indicating superior sensitivity of the COVID hunter over PCR. Additionally, on human subjects, we tested more than 1,000 human subjects with the COVID hunter. Our results showed that all COVID-19 confirmed cases were detected by the COVID hunter as positive, showing 100% accuracy. Interestingly, 4.7% of PCR negative, but are COVID positive. These are PCR false negatives. Human subjects were identified as positive by the COVID hunter, again showing superiority of the COVID hunter to detect patients who were diagnosed as PCR negative. Here is a unique application of the COVID hunter. Out of 76 out of 94 confirmed positive individuals. So these are confirmed positive individuals. They are infected with the virus. 94 tested. 76 out of those 94 were shown to be infective. How do I know they're infective? How do I know they're transmitting the disease? The virus was detected in their cough or sneeze or air exhaled from the mouth or nose and caught in a plastic bag or a plastic container. And we detected that immediately. So you make somebody cough in a plastic bag and you scan that bag. And you can detect the virus in air inside the bag or the container immediately. Demonstrating a unique feature of the COVID hunter to identify infective individuals or spreaders of the disease. Further, using the COVID hunter, thousands of objects were scanned for contamination with the virus, and contaminated objects were successfully detected and properly decontaminated. Of significant importance to the scientific community, especially to scientists and researchers working on COVID research. So those of you who are working on COVID research all over the world, listen to this. The ability of COVID hunter to track the modes of transmission of SARS-CoV-2 and its tolerance to environmental factors was tested. So we track the virus as it moves from hand to mouth to other people and goes from one object to another, and we subjected the virus to environmental factors, temperature fluctuations, and we measured that with the COVID hunter. Our team was able to show with high efficiency how the virus is transmitted from an infected person, so this is an infected person, while coughing into their hands, and then shaking hands with another person with clean hands. And that other person with clean hands touched a door knob. Then after that, touching a mobile phone, then touching a car. And we went with the virus as it goes from one object to another immediately, and we showed that this healthy individual, by shaking hands with an infected individual, can transmit the virus to a third party without even being infected. This is the hallmark of this virus. Well, this is why this virus is causing such pandemic, because it has a high rate of infection. And the only way to stop it is by tracking this and stopping it. So we are trying to stop it from going to a third party to a second party. Additionally, our team was able to discover the virus on the hands and feet of uninfected domestic pets. So these are animals, demonstrating how animals can be silent transmitters of the virus from person to person within the same household. So there was a dog that was going from child who's infected, touching, and going to an elderly and touching, and the dog, we detected the virus on the hands and legs of the dog, but the dog was not infected. In summary, Our results showed that the COVID hunter can immediately and accurately detect SARS-CoV-2 inside and outside the human body with high reliability and ease of use. I am personally excited to see the COVID hunter go from the current prototype phase to a fully refined, manufactured device that can be used to save lives around the world. This is the time to save lives and stop the spread of this pandemic and gain our lives back. I strongly urge my scientific colleagues in all fields of science and engineering all over the world to put the COVID hunter to the test. Put this device to the test and test it. And we need to work together as one team worldwide to advance our collective human knowledge about this virus in order to gain the war against this virus. With this, I conclude the presentation of our scientific data and research on the virus.